Hello students, I'm sure by now you have understood what is mitosis from the last presentation which was uploaded. Well, today we are going to continue with the chapter on cell division, and today's topic, as you can see, is the reduction division or meiosis. We all know that these are the two primary type of divisions that a cell undergo. But before venturing into the details of meiosis, we must understand some of the basic points of meiosis. You can see that historically it was Edward von Beneden who actually first discovered the meiotic cell division. However, its details and its significance were worked out by Weizmann at a later time. Now, meiotic cell division is a special kind of cell division that occurs only in sexually reproducing organism. Now, that includes plants, animals, microbes, fungi, and every living organism in which sexual reproduction is present and where gamete formation takes place. Therefore, it is obvious that meiosis or the reduction division is a prerogative of certain cells. We call them the germinal cells, which would be giving rise to the gametes. It does not occur in the other body cell or the other somatic cell. Now, the basic principle of meiosis is that it converts a diploid cell, diploid in terms of its chromosome number and chromatin value, into haploid germ cells, or in other words, gametes. Because as we know, sexually reproducing organisms produce haploid gametes which, when unite, that is, when fertilization occur, a complete diploid organism in terms of the embryo, the first cell, is restored. So that is one of the most important points to be remembered. Now, each daughter cell that are formed during meiosis has n number of chromosomes, which is known as the haploid number, because twice n is the diploid number and n is the chromatin number. And all sexually reproducing organism or all diploid organism have even number of chromosomes, that is, even pairs that are formed. As in our case, human being, we have got 23 pairs, but the chromosome number is 46. Now, in meiosis, we find that unlike mitosis, which is a straightforward process, and for our convenience we had made it into 4 distinct steps, meiosis is also a continuous process, but since it has got elaborate stages and there is complications, we have divided meiosis into 2 subparts, which is the first meiotic division and the second meiotic division. Within the first part, we have all the stages, the prophase, metaphase, anaphase, and telophase, and the same is true for the second meiotic division. Now let us see. The production of offspring, as I just said, by sexually reproducing organism includes formation of gametes and their fusion, and therefore you have already learnt it that gametes are specialized cells. Now they have to be haploid so that the diploid number is restored. Now, meiosis, as I said, ensures the process and keeps the continuity in sexually reproducing organisms. Now, as I was saying, that meiosis has got two stages, substages. One is the first meiotic division and the other is the second meiotic division. Now, the first meiotic division is very, very important, as because it is in this part that the entire genetic components realign themselves, they undergo exchange among themselves, and new combinations of genetic material within the cell is created. The reduction division or the reduction in number of chromosome actually occur in the first meiotic division. The second meiotic division is essentially mitotic in nature, where only the components are equally halved. We shall see to that at a later part of this presentation. Now, all the four subphases are present under meiosis I as well as under meiosis II, and these are common: the prophase, the metaphase, the anaphase, and the telophase. Yes, to recognize them, we have put the number 1 for the 4 stages under meiosis I and 2 for the stages under meiosis II. Now, here is a flow chart. If you observe it properly, the division meiosis has got these two subparts, meiosis I and meiosis II. The interphase part, of course, is present in all the cell irrespective of mitosis or meiosis, so we are not going into that. We have already enlightened ourselves in a previous presentation what interphase is and why it is important for any division whatsoever. Now we can see under meiosis there are 4 phases of meiosis I. Likewise, under meiosis II, there are 4 phases, and both of this meiosis I and meiosis II is followed by something called cytokinesis. But something to observe here, and is very interesting, is that the prophase I of meiosis I is again subdivided into 5 substages, which are known as the leptotene, the zygotene, the pachytene, the diploid, and the diakinesis. Often the pronunciation used is leptoteny, zygoteny. Both are correct. Now, after the prophase I is completed, it follows to metaphase I, anaphase I, and telophase I, and then the mother cell divides into 2 daughter cells, however, with n number of chromosomes. But the chromosomes are still in double the chromatin value that we are targeting. So this daughter cells, the 2 daughter cells so produced, enter the meiotic II division and undergo the 4 substages, ultimately becoming 4 cells, 2 from each. So we have got 4 cells at the end. Now we shall go into each of these stages one by one to understand and appreciate the way in which the process of meiosis, both 1 and 2, are completed. Now for prophase I, it is, as you can understand, is the longest phase in the meiotic division, and as you can see, it takes about 90% of the time. And I've already told you that there are 5 substages: the leptotene, zygotene, pachytene, diploid, and diakinesis. Now, what is leptotene? If you think of a cell that is a cell of the germinal epithelium and is present in the gonads, in case of plant, the ovary or the anther, then what happens? This specialized cell, which is going to divide and become a gamete, has already gathered its component during interphase. Now, during leptotene, we find that is leptotene is the prophase one of meiosis 1. We find that the chromosome becomes conspicuous, they condense, and they look like threads, fine threads. They start to become visible. Now, there's a marked increase in the volume of the nucleus. And after some time, we see that the chromosomes are starting to behave as if they are searching for their homologous pairs, because we all know that each diploid cell has got homologous pairs of chromosome. How many pairs will depend on the species, because in various organisms the number of chromosomes vary. That means the number of chromosome pairs vary. Now in zygote, what happens? Say we are talking about 3 sets of, or here in the picture you can see 4 sets of homologous chromosomes. But if you observe properly, you would see in each of the pair, one is red and the other is blue. Now this is for recognizing the paternal and the maternal chromosome in that particular cell. Now say this organism has got 4 pairs of chromosomes. The total number of chromosomes is 8, suppose. Like we have 23 pairs and total number is 46. Let us consider that this cell has got 4 pairs and 8 numbers of chromosomes. And for each pair, one is from the paternal side and the other is from the maternal side. Now what happens? This homologous chromosomes in zygote stage of prophase 1, they come to lie side by side, and this condition is called synapsis. To synapse is to adhere side by side. And when if I put 2 eyes side by side or 2 ones side by side, then I would say the 2 1 number are in a homologous dyad condition, because one stick would be called a monad, 2 of them placed together would be called a dyad. Now for this case, one stick is blue and the other is red. So in zygote stage, very specifically we have to remember that the homologous chromosome create synapses and they come to lie side by side. And there is a formation of synaptic bridge between the chromosomes, between the 2 homologues. Now what happens? In late zygote, we find that these 2 homologues, as many pair as there are, all of them, they start showing X-like structure. Earlier we had 2 pencils, one blue and one red, and suppose 4 sets of those. Each of this set of blue pencil and red pencil would now be a blue X and a red X. So what we have got 4 sets of blue X and red X pair. Now such a condition is called a tetravalent condition, because I told you the pencil was a monad. When 2 pencils were together, they were dyad, and now that there are 4 arms in one and again 4 in the other, so if we consider them as 4 different lines, then there are 4 tetrad. So we call it a tetravalent. Okay, a monad, a dyad, and a tetrad. A tetrad signifies 2 axes, where there are 4 total arms, because from top to bottom, that's 1 chromanimeta. So there are 4 chromanimeta. In 1x there are 2 chromanimeta, but 4 tetrad, 4 chromatid, and in 2x's there are 4 chromanimeta but 8 chromatids. Observe these words that I am saying. In 1 pencil, it was a monad. When 2 pencils are placed side by side, it is a dyad. But when one of those pencils becomes an X, it has got 2 chromanimata but 4 chromatids. When its partner pencil again becomes X, so the 2Xs has now 4 chromonemata but 8 chromatids. Remember these words. Now, this condition culminates the zygote stage. The next stage that we find the cell entering into is known as the pachytene. The pachytene is a very important stage because it is in this particular stage that the pair of X's, remember, in the pair of X's, one is from the mother and the other is from the father. So what happens? Now, the pair of X's starts putting their chromatids across each other. Now the blue X will put one of its chromatin over an arm of the red X, and maybe a red X's leg will cross with another leg of the partner blue X. So this formation of crisscrossing between and amongst the chromatids of the non-sister chromosomes, we find that there is formation of something called the chiasma. I would repeat this point. We are imagining two X's and one arm of one of the X, say the blue X, and one arm of the blue X has overlapped on the red arm of a red X. So the blue and red has formed a point of contact, and that point of contact is called the chiasmata. And it is at that point of contact that there will be breakage and exchange of the parts. That means the blue X will get a part of the red X and the red X will get a part of the blue X at the point of contact where there was breakage and reverse attachment. This is important to note that the breakage is there, but while there is attachment, the blue piece gets attached to the red piece and the red piece gets attached to the blue piece. So, during pachytene, we find that there is cross and exchange of chromatin material between homologous chromosomes and specifically between non-sister chromatids of the pair of homologous chromosomes. So what is happening? The maternal and paternal chromosomes are now losing their identity and are becoming mixture of each other. So the cell which belongs to a certain individual, and in that cell there was a paternal chromosome and a maternal chromosome, is now turning into a mixture of maternal and paternal chromosome. So this process goes on in the pachytene stage. Meanwhile, we observe that the centrosome, which is outside the nuclear membrane, had divided into two and is moving away towards the opposite pole, and at the same time spindle fiber, microtubular fibers are starting to form. In diploid, we see that at the point of contact, which I just named as the chiasma, there is a development of repulsion, and those homologous chromosomes slide on each other, thereby taking those exchanged part along with themselves. And this symptom or this process is called terminalization. Here what happens? The crossover chromosomes, they are now called the crossover chromosomes because they have exchanged parts, but while they have exchanged parts, they are now trying to push each other, and the X's are sliding above each other along the length of the chromatids. So when that happens, there is a physical pushing away from the partnership. Now that is specific to the diplotene stage, and we see that by the late diplotene, the spindle fiber has formed to quite an extent. The nucleolus is started to disappear, and the nuclear membrane also is starting to disappear but has not disappeared totally. Now the last stage under prophase 1 is the diakinesis. Now in this stage there is further shortening and thickening of the chromosomes, condensation takes place, nucleolus and nuclear envelope completely disappears, the spindle fiber and the spindle organization is completed, and obviously the centriole had migrated to the opposite poles so that they could hold the microtubules to a point of attachment from where the microtubules can come and get attached to the pairs of homologous chromosomes. Now that brings us to the end of prophase 1. Now we shall move into metaphase 1. Remember, we are still in the first meiotic division. Now in metaphase 1, a very interesting event occurs. If you observe the picture, you would see that all of those pairs of X's are not completely blue or completely red. They have now become red and blue in their own right. You cannot call them paternal chromosome or maternal chromosome. No, they are now crossover chromosomes. However, they are homologous, so they are together. Now what happens? In metaphase 1, the nuclear envelope is no more there. Some of the organelle is also no more there. The nucleolus is no more there, have dissolved in the matrix, and the crossover homologues, tetravalents, you can see in a pair of X there are 4 lines but 8 individual points. So we call in one X there are 2 chromanimata but 4 chromatids. So in a pair of X's there are 4 chromanimata but 8 chromatids. Okay. However, the homologous chromosomes, now we shall call them crossover homologous chromosomes, are standing in pairs, and all of these crossover pairs have come to lie in a straight line along the center of the spindle. Now from each end of the spindle, the microtubular spindle fiber will arise, which have already formed, and will attach themselves with the centromere of the respective chromosome on its size. The microtubule from the right side will attach the chromosome on the right side, and that from the left will attach with those of the left. But remember, they are always in pairs, and this is a situation which readies the cell to move from its double status to single status in terms of chromosome number. Let's see. Now in anaphase, the actual reduction of chromosome number is happening. As you can see in the picture, that during anaphase there is shortening and pulling exerted by the spindle fibers. As a result, the crossover homologous chromosomes are now separated from their friendship and are taken away towards the opposite poles. But if you look at each of these separated chromosomes, the X-shaped chromosomes, all of them are partially red and partially blue because they have already exchanged parts of themselves during which phase? During pachytene of prophase 1. Remember that. So this is the beauty of anaphase. However, you must remember that during anaphase, because of the pull, the X-shaped chromosomes don't remain stiffly as X. They would show L-shaped or V-shaped structure, double-armed L-shaped V-shaped structure, because they will be pulled towards the opposite pole. So naturally there will be a movement induced shape. All right. Now, in telophase I, once the crossover homologues have been separated and taken away to the opposite poles, the spindle fibers will disappear. New nuclear membrane will reappear. So also the nucleolus. And you can observe that earlier we had 4 pairs of axes. Now we have got 4 axes in one and another 4 axes in another. But this 4x in one and the other 4x in another is very different from the original 4 pair of axes. They were pure red, 4 were pure red and 4 were pure blue. Now what we have? We have 2 cells. The cell is forming. The furrowing is continuing to grow because of the microtubular central constriction, as I told you during mitosis presentation, that the cell is forming the furrow at the center. So 2 cells are being produced, and in case of plant cell, phlegmoplast wall will be formed, that you know. The 2 cells that will be formed now have haploid numbers of chromosome. From 4 pairs, they are now 4 each. However, they are crossover. They are mixture of paternal and maternal qualities in terms of genetic component. Yes, by the end of telophase, cytokinesis will occur and 2 daughter cells, as you can see, have been resulted. These 2 daughter cells are now N in their ploidy. That means haploidy or diploidy, these are the terms we use. So it is haploid. But remember, even though they are haploid in their chromosome number, the chromatin quantity is still 2C, because each of the chromosome has still a X shape. That means there are 2 chromanimeters. This is the stage because when the cell for meiotic division had started, from 2N 2C it has become 4N 4C. It has doubled during interphase. So now that we have just achieved our purpose of converting 4N into N, okay, the diploid value, which is 2N, has now become N, but the chromatin value has not yet gone down. So for that we need to go into the second meiotic division, and each of these 2 daughter cells, which are haploid in nature but are having N number of chromosomes which are in X shape, that means with double arms, have to become single armed. Let's see. The second meiotic division, as I have told you earlier, has also got all those 4 phases: pro, meta, ana and telo. Now the axes that you saw in the earlier slide, that each of them is in a dyad condition, because a single pencil is a monad and 2 together in X shape or 2 placed together is a dyad. So each dyad is composed of a pair of sister chromatids, as we know. In metaphase, centromeres are positioned at the equatorial plane. Well, in anaphase, these are separated. The individual participant of the X are separated. The X's become I's actually. And telophase, again the cell undergo constriction division. You shall understand this better with this diagram. This is meiosis 2. So we call them prophase 2, metaphase 2, anaphase 2 and telophase 2. And the prophase 2, we start from the cytokinesis of meiosis 1, where we had 2 daughter cells with haploid number of chromosomes, but all of them in X shape. In the earlier picture, we had 4 X's. Here, for our understanding, let us consider that the cells have got 2 X's and 3 X's. It's all the same, having 2 X's or 3 X's or 4 X's or 23 X's. Does not matter. So what will happen in metaphase? Those X's will be again pulled away from their junction, because the axis will align along the spindle fiber. Spindle fiber will be formed again. You can see in the prophase, spindle fiber will be formed again, and they will connect with the center of the X of each of those multicolored X, and will pull one part of the X towards the other side and the other part towards the other side. That will happen during metaphase and an anaphase of meiosis II. So once that happens, now by late anaphase and early telophase, the cell will start constricting. Now that the cells are in true condition, having haploid number of chromosome, you can see 3 in one side and the 3 in the other side. In anaphase 2, both in the upper picture and in the lower picture, 4 cells are forming, but each of them has got only 3 chromosomes, and all the 3 chromosomes are to some extent combinations of blue and red. Now there are 2 in which there is no blue and red, only red and one only blue. Well, they did not undergo crossover. It is not mandatory that all chromatids will undergo crossover. They might not. Or any number can go double crossover, triple crossover. That is also possible. This is just for understanding that the crossover chromatids are now separated from their X shape into eyes and are taken away into 2 and 2 daughter cells, because earlier in meiosis 1, 2 daughter cells were produced. So these are the 2 daughter cells. From these 2 daughter cells, we have ultimately got 4 daughter cells, which are purely 1N, 1C. In terms of chromosome number, they are haploid, that means N. If it is human being, we will have only 23 chromosomes, 23 only, number, not pairs. Here only 3 in each of the cells. And the chromatin value is also 1C. That means our total chromatin value is 3 picogram in human beings. It will be 1.5 picogram in quantity. So the quantity also will be halved. The number of chromosomes will also be halved. Now these cells are the specialized cells which we call the gametes. Now there could be the male gametes, the sperms, or there could be the female gametes, the ovas. Because in a sexually reproducing organism, a male gamete has to unite with a female gamete. So if there is no reduction in the number and quantity of these cells earlier, then every generation will give rise to a new animal organism, because the chromosome number will not remain constant. It is meiosis which is really keeping the species constancy. From 23 pairs, the gametes become of 23 only. So when 2 gametes meet, again the 23 pair is restored. The chromatin value is restored to 2C. The chromosome number is restored to twice N, and it is the embryo for a human being. Likewise, for all sexually reproducing organism, 2 haploid cells having N number of chromosome and C quantity of chromatin are resulted, which do no other function in the body. They have got no other function. If they are wasted, they are wasted. But only and only if they come together, a new life is formed. So, meiosis maintains species constancy. Well, there are other important aspects of meiosis too. Now, if we look into the functions of meiosis, of course, production of haploid gametes so that fertilization restores the somatic number and the diploid organism is recreated. That's one very important point. But another very important point is, it is because of meiosis that there is variation, because crossing over is happening, exchange of genetic material is happening between the paternal and maternal chromosomes. So what we are getting is a constant changing of the qualities, and this is the raw material for mutation, therefore variation, and in the long run, evolution. What we are looking at is a process of constant change. We might not recognize it in our lifetime or even in hundreds of years, but over millennia, even we human beings are sure to change from what we are today into something else, because we have changed from what we were to what we are today, and we will change surely, and that is a 100% guarantee, from what we are today to something else, because the raw material is working constantly, and with every generation a new variety is being created. So very subtly but surely changes are being incorporated. Add to it the mutational changes that occur suddenly, and what you have is a perfect concatenation for variation, isolation, speciation, evolution. Okay, so that's how meiosis is important to entire living organism, the living world per se. I hope you have understood that the importance of meiosis and the processes. I'll just sum up very briefly that meiosis is the process of reduction division, where a germinal cell divides from a diploid condition to a haploid condition, where the chromatin value reduces to C from 2C, and we get the gametes. The process is a continuous one but is divided into two parts, meiosis 1 and 2. Both these parts have got prophase, metaphase, anaphase, and telophase. We mark them as 1 and 2 for both the two parts. But under meiosis 1, under prophase 1, there are 5 substages, which are leptotene, zygotene, pachytene, diploid, dikinesis. Now these are the very, very important part, because the actual case of exchange of material occur in the pachytene stage of prophase 1, where the parental chromosomes share parts among each other and become crossover chromosomes. So that is the beauty of gamete formation and the meiotic cell division, which is very specific, meant for a certain type of cell, but is supreme in its quality of maintaining the species constancy and of continuing the life and generation of sexually reproducing organisms. Now, the cell is the structure and functional unit of life. You have everything in it, we have everything from it, and it is from this cell that we are. Thank you.